Cleanroom Fundamentals. What is a cleanroom? Hello and welcome to Chapter 1 of our Pharmaceutical Cleanroom Training Series. In this chapter, we will build a practical understanding of cleanrooms, classification systems, airflow, pressure control, environmental monitoring, and personnel practices. Before we begin, please subscribe to PharmaTalks and watch the rest of our educational videos so you do not miss any of the specialized pharmaceutical training in this series. If you would like the file used in this video, the complete educational file collection or information about our related courses, leave a comment below and we will send you the relevant details. So what is a cleanroom? A cleanroom is a controlled environment where airborne particles, microorganisms, temperature, humidity, and pressure are maintained within defined limits. Its purpose is to protect the product, the manufacturing process, and ultimately the patient. 4 core purposes. A cleanroom serves 4 core purposes: protecting the product, protecting the process, protecting the operator, and meeting GMP requirements. Contamination control helps preserve the quality and safety of pharmaceutical products throughout manufacturing. The facility design and operating procedures may also protect personnel from hazardous materials. Together, these controls support compliance with good manufacturing practice. Classification systems. Cleanrooms are classified using recognized standards that define their level of air cleanliness. EU GMP uses grades A through D, while ISO 14644 commonly identifies environments from ISO Class 5 to ISO Class 8. The main classification metric is the concentration of airborne particles of specified sizes within a defined air volume. In general, a lower ISO class number represents a cleaner environment with stricter particle limits. Airflow patterns. Cleanrooms mainly use 2 airflow patterns: unidirectional or laminar airflow and non-unidirectional or turbulent airflow. Laminar airflow moves in a controlled and nearly uniform direction, helping carry contamination away from critical areas. Turbulent airflow mixes clean supply air with room air to dilute contamination before it is removed. The correct pattern depends on the room grade, the process, and the sensitivity of the product. HVAC, the heart of the cleanroom. The HVAC system is the operational heart of a cleanroom because it controls air quality and air movement. Fresh air passes through a pre-filter, is conditioned by the air handling unit, and then moves through a HEPA filter before entering the room. The system provides filtration, the required air change rate, and stable temperature and humidity. A failure in any part of the HVAC system can directly affect cleanroom classification and performance. Pressure differentials. Pressure differentials between adjacent rooms form an important barrier against contamination. In many pharmaceutical areas, the cleaner room is maintained at positive pressure so that air moves toward the lower grade space when a door opens. A typical differential between adjacent rooms is around 10 to 15 pascals, although the exact value must follow the facility design and risk assessment. Continuous monitoring helps detect any loss of the intended airflow direction. Typical environmental conditions. Temperature and humidity control support operator comfort, product stability, and reliable process performance. Many cleanrooms operate at approximately 18 to 24 degrees Celsius and 45 to 60% relative humidity. These are general ranges, and actual limits must be based on the product, process, and site requirements. Continuous recording, alarms, and trend review help identify undesirable changes before they become serious deviations. Gowning sequence. Cleanroom garments must be put on in a defined and carefully controlled sequence. Jewelry is removed and hands are washed and dried before the suit and hood, mask, shoe covers, and sterile gloves are applied. During gowning, clean garments must not touch the floor, walls, or other potentially contaminated surfaces. Following the correct sequence minimizes the transfer of particles and microorganisms from personnel into the cleanroom. Controlled entry and exit. Personnel should enter progressively from areas of lower cleanliness toward areas of higher cleanliness. During exit, the same route is followed in reverse to reduce contamination transfer between zones. Interlocked doors must never be opened at the same time because this can disturb the pressure cascade and airflow direction. Entry and exit routes should also be managed to minimize crossing and unnecessary movement. Environmental monitoring. An environmental monitoring program demonstrates whether a cleanroom remains within its defined state of control. It typically covers airborne particles, viable air, surface microorganisms, temperature, relative humidity, and differential pressure. Results must be recorded, reviewed, trended, and compared with established alert and action limits. Trend analysis can reveal gradual deterioration before it develops into a major deviation. Microbial control methods. Several complementary methods are used to assess microbial contamination. Active air sampling, settle plates, contact plates, and swab sampling each provide information about a different aspect of the environment. Sampling locations, frequency, and exposure times should be selected according to process risk. Alert and action limits must be predefined, and every excursion requires documented investigation and appropriate follow-up. Common contamination sources. People are often the largest source of particles and microorganisms in a cleanroom, especially when gowning or behavior is inadequate. Materials, equipment, disrupted airflow, poor cleaning, and open doors can also introduce or spread contamination. Inadequate HVAC maintenance creates an additional risk by affecting filtration, pressure, and environmental control. An effective contamination control strategy must identify and manage all of these sources as one integrated system. Material and personnel flow. Material and personnel routes should be separate, one-way, and non-crossing wherever possible. Personnel use dedicated entry and exit airlocks, while materials are transferred through material airlocks or pass boxes. This separation reduces contamination transfer and prevents the return of items from dirtier zones into cleaner areas. Every route should be supported by clear signs, written procedures, and effective training. Effective cleaning and disinfection. Effective cleaning and disinfection involve much more than selecting a chemical agent. A written procedure, an approved product, the correct concentration, sufficient contact time, and the proper application method are all essential. Disinfectant rotation should be based on risk assessment and validation evidence. Trained personnel must perform and fully document every activity to maintain traceability. The Cleanroom Golden Checklist. To conclude this chapter, a successful cleanroom depends on several connected controls: controlled air, correct pressure, disciplined personnel, continuous monitoring, validated cleaning, and complete documentation. A weakness in any one of these areas can affect the performance of the entire system. A cleanroom should therefore be understood not simply as a physical room or a collection of filters, but as an integrated technical and management system. When these principles work together, they protect product quality, process reliability, and patient safety.